Recent advances in genomics and information technology have started to have a major impact on biomedical research and patient care, in particular in the field of oncology. Many technical challenges must be addressed, including the integration of clinical, genomic and imaging data on a large scale. To address these challenges, the Integrate project, funded by the European Commission, has developed an innovative infrastructure and tools to enable data integration, data sharing and large-scale collaboration. 7 partners, with a range of expertise from clinical research coordination to healthcare IT systems development, have participated in this project.
The Integrate project has been driven by clinical needs in cancer research and care with a primary focus on breast cancer. Several tools have been developed to address specific needs, but the underlying IT framework that we've built in this project is generic and addresses important aspects of clinical data sharing and exploitation. The first aspect is semantic interoperability. We have designed a system that can integrate complex medical data from various sources and represent it in a uniform way. This makes it easy to build collaborative tools to share and explore clinical data. The second aspect we have tackled is data security. Researchers and patients are willing to share clinical data, but only if they are sure that the proper safeguards are put in place. The security framework that we've built and Integrate tackles all these complex requirements of modern, collaborative clinical care and research. And finally, this project has strongly focused on testing and validation of the tools that we've built.
Decima is a tool allowing clinicians to automatically match patient data with a database of clinical trials eligibility criteria.
The advantage provided by a comprehensive cancer center like Institut Jules Bordet is the availability of many clinical trials. So in daily practice, we try to enroll patients in these trials whenever we think this might be beneficial for them. With the recent advances in genomics, such as tumor DNA sequencing, these trials are targeting smaller groups of patients with specific molecular aberrations. A consequence of that is that matching patients with clinical trials becomes more complicated and more time-consuming. The Decima patient screening tool makes this easier by retrieving the patient clinical data from the electronic health record and comparing them with the eligibility criteria from trial database, which can potentially be very large. The tool also highlights the criteria for which some information is missing or the matching is inconclusive, requiring some manual verification. So overall, this tool can make patient enrollment both easier and faster.
Nona is a tool allowing a clinician or a researcher to easily select cohorts of patients from a research database.
In a research setting, one frequently needs to gather statistics from a subset of patients. For example, it might be interesting to select all breast cancer patients who have received a certain treatment and have responded well to that treatment. Or one might want to evaluate a proportion of patients who have developed an adverse reaction following the administration of a specific drug. Another application of the tool is trial feasibility. One of the early steps in the development of a clinical trial is to determine if one will be able to recruit enough patients to run the trial based on the characteristics of the patients. With Nona, this can be done easily and quickly thanks to the graphical user interface. One major benefit of Nona is the possibility to do cohort selection automatically on an integrated database with multiple datasets, which greatly facilitates meta-analysis. Another nice aspect of Nona is that it allows to collaborate by sharing in a controlled manner the filters and queries that have been previously defined. Overall, the Nona tool will allow to perform research more rapidly and more efficiently and enhance collaboration.
The Integrate Central Pathology Review platform enables the collaborative review of pathology slide images in the context of clinical trials.
One of the main problems we are facing in cancer pathology is the variability in the way different pathologists score certain characteristics of the tumor, particularly between laboratories. So the way we have been addressing this problem has been ring studies, where the same pathology slides are exchanged between the different laboratories and scored independently. This is logistically complicated, time-consuming and costly. The Integrate Central Review of Pathology tool facilitates the process tremendously. It allows uploading of high-resolution scanned pathology images from multiple labs and remote visualization and annotation of the images by a group of experts at different places and different times. It also makes it very easy for the coordinator of the study to compare the different results and find potential problems.
The Integrate Analysis platform supports visualization and statistical analysis of the clinicogenomics data, which is stored in the Integrate platform.
The idea behind the analysis platform is to support visualization and statistical analysis of the clinicogenomics data, which is stored in the Integrate platform. One of the advantages of this tool is that it is user-friendly and can be used by people who have some experience of clinical and genomic data analysis, but not necessarily a deep knowledge of bioinformatics and biostatistics. The type of analysis that can be performed goes from simple descriptive statistics and plots to much more sophisticated analysis, such as survival analysis, comparison of treatments, or even multi-scale predictive modeling to predict drug response and assess candidate biomarkers in groups of patients. The tool also allows to document an analysis pipeline and the parameters and data that have been used for a specific analysis. This is important for the reproducibility of research. Another advantage of the tool is that it is coupled with the Integrate cohort selection tool and the semantic layer. This allows to rapidly perform meta-analysis by selecting patients across studies and extracting the relevant information.
For more information on the Integrate project, please visit www.fp7-integrate.eu.